Hello everyone, I hope you are doing well. Today we are going to cover development of the limbs, and the scope for the session will be to describe the tissue of origin of the limbs, also describe the very important signaling centers of the limbs, the purposes of exams, also describe the embryological development of the limbs, and then we'll check on the congenital anomalies of the limbs. So for the tissues of origin of various structures that are formed on the lower limbs, so the surface ectoderm and the paraxial mesoderm and somatic layer of the lateral plate mesoderm will give rise to the various derivatives of the limbs. So I'll take us back to the three germ layers. So the three germ layers usually have the ectoderm, usually have the mesoderm, then usually have the endoderm. So the mesoderm will divide into three. We'll have the paraxial mesoderm, the mediate mesoderm, and then the lateral plate mesoderm. Lateral plate mesoderm divides into two, somatic and splachnic, now regarding to what they are covering. For the ectoderm, it will, when we form the neural tube, that is the neural ectoderm, the remaining portion of the ectoderm becomes surface ectoderm, which will give you the skin, the epidermis of the skin. So at this point, what we're talking about is that the surface ectoderm will give us the epidermis and its appendages. So the epidermis and its appendages, when talking about appendages, we're talking about all the sweat glands that will be located in the limbs, all the sebaceous glands that will be found on the limbs be coming from surface ectoderm of the limbs. Then for the paraxial mesoderm, it will give us the limb musculature to talk about the muscles, the extensors and flexors of the limbs, and also the intrinsic muscles of the various limbs. Then the somatic layer, so this is somatic layer of the lateral plate mesoderm will give us the appendicular skeleton and the dermis of the limbs. So these are very illustrative image showing us how now the innervation of the muscles will happen. So we'll have the somites. We'll have the somites, which will migrate together with the innervation to go and form the various muscles. For the various signaling centers of the limbs, we usually have four signaling centers. One of them is the apical ectodermal ridge. So this is the apical ectodermal ridge, the AER. So the AER is actually found at the boundary between the dorsal and the ventral point of the limb. So the apical ectodermal ridge ideally controls the proximal distal growth, given the fact that it's found on the tip of the limbs. So when it controls the proximal distal growth, what happens is that prevents this zone of proliferation from differentiating. But in addition to that, it allows continued mitosis, and through that now you have elongation of the limb. What about the zone of polarizing activity? The zone of polarizing activity ideally comes from mesenchyme. This apical ectodermal ridge, it's ectodermal, so it's ectodermal in nature. Apical ectodermal ridge is called ectodermal, so it's ectodermal in nature. This zone of polarizing activity comes from the mesoderm, the mesenchyme, from the mesenchymal cells. So the zone of polarizing activity controls the anteroposterior axis. So it's the one that determines which finger will become the thumb, which finger will become the digits, and the small finger, which toe will become the big toe, which will become the small toe. That is the anteroposterior axis by zone of polarizing activity. But the other one, the dorsal and ventral ectoderm. The dorsal and ventral ectoderm comes from, ideally, they usually determine which side will become the dorsum, which side will become the palmar, or maybe the plantar aspect of the hand or foot. So, of course, at this point, you know that the dorsum is actually the upper part of the foot. Then the plantar aspect is the lower part, Then the palmar aspect is on the hand, then the dorsum also on the hand. So same aspect will realizable dorsal and ventral aspect. So the dorsal dorsum, ventral ectoderm will determine which side will become the palmar and the plantar aspects. You can now narrow down to the development of the limbs. So what will happen is that you'll have presence of the limb bars appearing on the lateral wall. So this is the lateral wall, you'll have presence of the various limb bars. So you'll appreciate that the limb bars, the upper limb bars will appear earlier than the lower limb bars. The upper limb, the lower limb bars. So they will appear on the lateral body wall, as seen in this. Then under the role of apical ectodermal ridge, we'll have elongation of the limb bars. So now you elongate, you'll have like continued growth, but it will reach a point when the effect of apical ectodermal ridge on this zone of proliferation won't be active. And due to that now, cells that are away from apical ectodermal ridge will differentiate into cartilage and then and muscle. So remember, cartilage and muscles are coming from somites. So at this point, we are having the apical ectodermal ridge. So it will lose its activity when it moves away from the proximal tissue. So through that, now we'll allow differentiation to happen. So through that, the limb will differentiate into three zones. So three components, that is the stylopoid, zygopoid, and autopoid. The stylopoid, which is here, will give us the humerus and the femur of upper and lower limb respectively. Then the zygopoid is a zygopoid. The zygopoid will give us the radius and ulna in upper limb, and then the tibia and fibula on the lower limb. Then the autopod will give us the musculature, the bones, the skeletal bones of the hands and the foot. So we'll talk about the carpal bones, metacarpal digits of the upper limb, and then tarsals, metatarsals, and digits of the lower limb. So that is how now you have elongation uh, due to apical ectodermal ridge, but now cells that are away from the apical ectodermal ridge, we lose stimulation, and that's the reason why they will differentiate the three components, which is stylopoid, zygopoid, and autopoid. So due to that now, they'll differentiate to cartilages. So how will you form the cartilages? The mesenchyme in the bud will condense. So this mesenchyme will condense. And then the mesenchymal, remember mesenchymal cells are undifferentiated cells. So these mesenchymal cells will differentiate to form chondrocytes. When you form chondrocytes, ideally that's the cartilage. So in the sixth week, we'll have cartilaginous models, hyaline cartilaginous model. Then after that, in the 10th week, the bones start developing by endochondral ossification. So remember, hyaline cartilage will give rise of bones via endochondral ossification. Then the 12th week now, you'll have primary ossification centers happening, and they start developing from the diaphyses. So primary ossification centers will start developing from the diaphyses, then to the epiphyses in the long bones. So remember the epiphyses are the ends, and then diaphysis is the shaft. Then secondary centers will form on the epiphyses. These are secondary centers will form on the epiphyses. Then you can also appreciate in these images how blood supply is very important in this process of ossification. So this comes across, and that's the reason why growth usually happens even after birth. We have epiphyseal growth plates, which will enable the elongation of bones. So how do you develop the joints? The various joints of the limbs will be formed by arresting of chondrogenesis. So remember, a good example, for example, the femur, tibia, and fibula will be formed by endochondral ossification. So in between, you'll have arrest of chondrogenesis. So when you have arrest of chondrogenesis, the cells around that region will proliferate. So when they proliferate, they form an interzone. So that interzone will be formed between the long bones. And through that now, how will you develop now a joint cavity? A joint cavity will happen by the interzone now, the cells ideally undergoing apoptosis, cell death. Then the joint capsule surrounding the joint cavity is formed from the surrounding mesenchyme. Then the mesenchyme on the ends of the bones forms the articular cartilage. Remember, the mesenchyme on the ends of the bone is cartilage. It's hyaline cartilage. That's the reason why it will form articular cartilage, synovial membranes, and ligaments of the joints. It is very important. We need to appreciate that on how the various joints are formed. So you have arrest of chondrogenesis, then the cells around those bones will proliferate and increase in density, creating an interzone. Then the cells in the interzone will undergo apoptosis, forming a joint cavity. Then the mesenchyme surrounding this joint cavity will form the joint capsule. Then the mesenchyme on the ends of the long bones will form the articular cartilage, synovial membranes, and the ligaments of various joints. How then do you form the hand and the foot? So the hand and the foot, remember, will be formed on the distal aspects of the limb bud. So we have development of the limb buds. How will development of the limb buds occur? Limb buds, remember, they occur during embryology the fourth week. So they continue to elongate, but now after you have differentiation of uh, the, um, the zygopod, the stylopoid, the autopod now will come and form the hand and the foot. So we'll have development of the limb buds. For example, now this A is for the upper limb bud, G is for the lower limb bud. So what will happen is that we'll have limb bud flattening. So the limb bud will flatten to form foot or hand plate. So this is the foot plate, this is the hand plate. The paddle-shaped hand on the foot plates. And what will happen is that we'll have mesenchymal condensation. The mesenchyme will condense to form 5 digits. So mesenchymal condensation will give you the digital rays. So you can appreciate that they have mesenchymal condensation, but you still have a membrane covering the digits, the mesenchymal condensation. Then you'll have notching of the digital rays. Notching will happen. And then finally, what will happen on this notching is that the membrane will undergo apoptosis to give us a hand or a foot. So this is a hand and a foot after now the webbing happening and the apoptosis to ideally continue. So when you have apoptosis, you have your hand and the foot plates. How then do we develop the limb musculature? So limb musculature, as we talked about, is that they migrate from the somites. They form from the somites that migrate into the limb buds that form muscles. So what will happen is that the somites that are just next to the vertebra, the neural tube, will migrate together with the innervation, and then they will divide into two compartments, flexor and extensor compartments. So what will happen when these muscles will divide into flexor and extensor compartments? That's the reason why most of the flexor muscles and extensor muscles, they usually have same innervation. Then these muscles will either divide further or fuse form individual muscles. So for example, when you want to form the various extensor muscles, you will further divide the extensor compartment to give you the extensor muscle. Same case with the flexor muscles. These are very important image showing us how the hypomere will form the various limb musculature. You can appreciate the innervation is actually coming from the ventral rami. So the ventral rami innervates the hypomeres. Remember the hypomeres will give us the muscles of the trunk, that is anterior abdominal wall, muscles of the thorax, muscles of the limbs, and muscles of the neck. But now for the muscles of the back will be coming from the epimeres, which will be a topic for discussion another day. So supplied by the dorsal rami. But primary rami, ventral primary rami supplies muscles of anterior abdominal wall and also the muscles of the limbs. So here is a question. Maybe you can go through it together. What are the differences on the development of the upper and lower limb? In terms of innervation, the upper limb pad is always ahead by 2 days. Location, of course, you know where the location of the upper and the lower limb pads. For the innervation, of course, we'll have brachial plexus for the upper limb, and then lumbosacral plexus for the lower limb, then angle and side of rotation is that you'll have 90 degrees medial rotation for the lower limb, and then 90 degrees lateral rotation for the upper limb. So those are the various anomalies. But we'll have images on them. So we can discuss these anomalies at this point. So this one is clubfoot or talipes, congenital talipes equinovarus has anomalies. So you can appreciate that the leg at this point is inverted and adducted. So it has anomalies to do with the talus bone. You can appreciate this for familia, absence of intermediates. You can appreciate this person has polydactyly. So polydactyly is due to formation of more digital rays. So more than 5 digital rays. At this point, you can appreciate this is syndactyly. So there was absence of webbing, absence of apoptosis in this. And you can appreciate this is amelia. So absence of 2 limbs, 2 upper limbs are absent. Then all the 4 limbs are missing in this image, showing us this tetramelia. And so at the end, you can appreciate this is the two images that following actually one another are due to amniotic band syndrome. But now this is leading to limb constrictions. These are limb constrictions or the leg constrictions. If the constrictions can be worse, they lead to digital amputations or even leg or hand amputations, as we'll see in the next slide. Then on this other one, you can appreciate this is osteogenesis imperfecta, a disease that has to do with the hyaline cartilage defects. Then this is erythrogryposis multiplex congenita, erythrogryposis multiplex congenita, a disease affecting joints, stiffening of joints, such a way that the joints can't move. And then this one is digital. Also, this is digital amputation. And then this is limb amputation, as you can appreciate there. On this other one, ideally it's just a repetition of the same. You can appreciate this is polydactyly. This is for familia. You can appreciate this amelia, absence of a limb bud, absence of a limb. This is polydactyly, more than 5 fingers. This is brachydactyly. So brachydactyly, these are short fingers or digits. Then on this other end, you can appreciate absence of the radius bone. So this is radial agenesis. So remember, radial agenesis is a spectrum of disorders giving us various bones, as giving us like absence of radius bone. So the spectrum ranges from 1 to 4, with the most severe one being radial agenesis, like complete absence of the radius bone. So it's a spectrum of malformation known as radial ray agenesis. On this point, we can appreciate this is claw feet, then this is clubfoot, then of course it's polydactyly. Similar case we can appreciate in this one. This is claw hand, then this is polydactyly, and this lobster claw hand. So I can give us we go and check a question, an assignment, we go and check the differences between claw hand and these lobster claw hands. So we can appreciate this lobster claw hands, and what's the difference between the lobster claw hand, and then can also appreciate what is the difference between lobster claw hand and claw feet. In addition to the formation of mesenchymal digitations being formed. This one you can appreciate this is syndactyly. And for syndactyly, we usually have two types of syndactyly. You can have osseous syndactyly. So in osseous syndactyly, we have the fusion of the bones forming the various digits, but then you can also have cutaneous. So in cutaneous, only the skin is fused, no bone fusion. So cutaneous fusion, cutaneous syndactyly, only the skin fuses. On the other one, osseous syndactyly, you have the bones of the various digits fusing. On this other end, you can appreciate this is achondroplasia, an anomaly that affects the hyaline cartilage. So ideally, long bones will be formed properly. Then you have congenital displacement of the hip joint. Congenital displacement of the hip joint. At least one you can appreciate that there's stability with some missing, and also the head of femur is displaced from its location. So congenital displacement of the hip joint. And this one you have radial agenesis, so you can appreciate that a small bit of the ulnar bone is present, but the radius bone is being. So this is radial agenesis. So that is it. Um, I hope there is no questions. Thank you for joining in, and have a blessed time. For any questions, can you reach out? We'll be there to help you. And can you like, share, and subscribe to the YouTube channel.